In this video, we're going to talk about lipids, which is the last of the major 4 classes of biomolecules. In Chapter 1, we covered the first 3 of the 4 major classes of biomolecules: proteins, carbohydrates, and nucleic acids. Now we're going to cover lipids. Lipids include fats, waxes, and other related organic compounds that are not soluble in water. Fats are esters of glycerol and 3 fatty acids. Glycerol looks like this. So a fat will have an ester at each of these positions instead of an alcohol. Phospholipids are also esters of glycerol. However, only 2 of the positions form esters, and the last position would have a phosphate group instead of an ester. Waxes don't have a great definition. We know that they are malleable organic compounds and that you can manipulate them without them cracking or breaking on you. We also know that they are solid at room temperature, and they typically have long alkyl chains, but there aren't really a lot of things that you can look for structurally except for the long alkyl chain. Steroids have a specific ring structure derived from squalene. You can see the ring structure shown here with the A, B, and C rings each having 6 carbons, and the D ring having 5. Let's look a little bit more at fats or triglycerides. You can see glycerol shown here on the left. Glycerol has 3 alcohol groups. When glycerol reacts with a fatty acid, it can form the fat or the oil, as shown here on the right. Fatty acids have a carboxylic acid functional group and then a long hydrocarbon chain on the end. And we'll look at some examples of fatty acids on the next slide. Here in red, you can see the atoms that come from glycerol. And here in blue, you can see the atoms that come from the fatty acids. The R groups attached to the esters here may be different depending on the fat. Animal fats tend to be solid at room temperature. One good example is lard, whereas plant fats tend to be liquids at room temperature. So we usually consider plant fats to be oils. Here are some examples of common fatty acids that you might see reacting with glycerol to form a fat or oil. Fatty acids that contain single bonds, like lauric acid, palmitic acid, and stearic acid, are associated with fats that are solid at room temperature. Coconut oil is one of the few plant fats that is solid at room temperature. Fats that have double bonds like oleic acid, linoleic acid, and linolenic acid are going to be found in oils that are liquid at room temperature. Remember when we were talking about melting points and boiling points, and we said that increased intermolecular interactions increases melting point? These first 3 fats here have higher melting points because they have better intermolecular interactions in that carbon chain. Those carbon chains can stack nicely and thus get a lot of interaction. Once you throw those double bonds in there, it kind of puts a kink in the chain and decreases the number of intermolecular interactions. This decreases the melting point, and that's why these fats down here are liquid at room temperature. Phospholipids are structurally very similar to fats. Again, they have that glycerol backbone. They also have 2 esters from fatty acids. The only difference here is instead of that 3rd ester, they have a phosphate group. That phosphate group is usually connected to another molecule. In phosphatidylcholine, for example, the phosphate is connected to a choline fragment. The phosphate group is ionic and thus water-soluble or hydrophilic, and the long carbon tail is hydrophobic. So phospholipids can be used to form phospholipid bilayers. An example of a lipid bilayer is shown here. You can see the phospholipid with the polar head in red pointed towards the aqueous or water environment, and the long carbon chains pointed towards the middle. That makes this middle portion of the lipid bilayer nonpolar or hydrophobic. You can see the lipid bilayer in the cell membrane here, and there are many mechanisms to aid in the transport of molecules through that lipid bilayer. One of the most famous waxes is beeswax. You can see here a principal component of beeswax. Beeswax is of course malleable, meaning you can kind of move it around. It doesn't crack as you move it around. And you can see that this molecule here has a long carbon-hydrogen chain on both sides. Beeswax is also solid at room temperature. Lastly, we have steroids. As I mentioned before, steroids have this common A, B, C, D ring pattern. Steroids are derived from squalene, which is shown down here. And squalene is made up of what we call isoprene units. Isoprene is shown here. It has 1, 2, 3, 4, 5 carbons. And so this particular pattern and number of carbons is known as an isoprene unit. The isoprene units down here in squalene are highlighted for you. The squalene reacts to form this common structure, and that common structure is used to form several different types of steroids. Here are several examples of steroids. You can see that the only thing they really have in common is this ring pattern. Some of them have alcohols, others have carbonyl groups. Over here in estrone, we have an aromatic ring. Cholesterol has a carbon-hydrogen chain, whereas testosterone has an alcohol and estrone has a carbonyl group. So basically everything off that ring can be interchanged. But as long as you have that same ring pattern, then you have a steroid. And if you wanted to, you could go through and identify those isoprene units in all of these steroids. This wraps up lipids and concludes Chapter 2.